Hello. So this lecture is to go through parts of chapter 26, which is about cancer and cell biology. So let's talk about what is cancer. And cancer is really just uncontrolled cell division. So cancer is a disease of cell cycle. When cell cycle is no longer controlled and all the checkpoints aren't met, you can get cancerous cells. And one of the things most clearly recognized about cancer is a tumor. Okay? So a tumor is just this mass of tissue. And if you looked at it microscopically, you would see that it's disorganized as compared to normal growth. The cells have different shapes, which we call morphologies. And we're going to talk about why these cells can do this, how they can get disorganized and pile up on each other, because normal cells don't do that. A couple other terms that we use to describe these tumors or these growths are benign, which simply means that tumor is going to stay in place. So it's not going to spread. And many times that means we can remove the tumor, but sometimes you cannot remove even benign tumors, and sometimes they can cause issues by putting pressure on tissue or other organs. So benign does not mean totally safe. And then we also have malignant tumors, which means the cancer can actually spread to different locations in your body. And these are the ones that we worry about the most. If you can remove a benign tumor, then you've removed the cancer. But if you have something that's making a tumor and able to spread to other parts of the body, then that's much more hard to control and hard to get rid of. The other thing we'll talk about in a second is you see this term differentiation, which is kind of hard to see in this image, but you can see as the cells grow and mature, they differentiate. They're actually moving, they're getting pushed up through the layers of your skin, and differentiation means that they mature into very specific cells. And in our skin especially, then we shed dead cells. So the reason we'll have normal growth in our skin is we have this balance between the dead cells and the newly being produced cells. Whereas with cancer, you have excess of new cells, not enough cells dying off, and that's where you get these massive growths. I am not a histologist, I'm not a pathologist, but I thought these couple of pictures kind of illustrated some of the abnormal characteristics of cancer cells. So you can see disorganization here comparing these two. At the microscopic level, you can see that the cells look different. They have different shapes. Here they have large nuclei with irregular shapes. Here's some normal cells. This is actually from a pap smear, so many women will go in every year or every couple of years and get cells from their cervix scraped, put on microscopic slides, and looked at under the microscope, and they're looking for abnormal cells that could suggest there's cancer. That's what they do in a biopsy. They remove some of your cells, some of the tissue, and they compare it to what normal tissue should look like. Okay. So you're not going to be identifying any of this, but I want you to kind of understand this phenotype of cancer and why it's an issue. If you're producing cells in disorganized tissue, the tissue is not going to function correctly. If you're building tumors, again, it's going to put pressure on tissues and organs and cause disease. So normally cells differentiate, and all that means, I kind of like to think of it as getting a job or becoming an adult, right? Many of you are already adults, but you may not have specialized, right? So cell differentiation, you can also call specialization. So we have cells in our body that are called stem cells. They're like, we call them like the baby cells, like, I don't know what I want to be when I grow up. And these can continually divide. And they can produce, depending on the stem cell type, all different types of cells. Okay. So their fate hasn't been determined yet. But once a cell gets that information, okay, you're going to become intestinal, or you're going to become blood, or you're going to become, up here, a grown-up skin cell, then with cell differentiation, many cells lose the ability to continuously divide. So now you've put kind of a limit on the life cycle of a cell. And that's important because we want this balance of new cells and dying cells to keep our tissue refreshed and healthy. We don't want bulky buildup of cells. So cell differentiation is very important, and now these all have unique functions. Many cancer cells regain this ability to continually divide, which can be a problem, right? If you want to have cells stop dividing, cancer cells don't always have that regulation. The key here is telomeres. So this is the key to why cells stop dividing. Okay, so assuming everything else is normal, a cell will divide about 50 times. Right, division, right? And this is called the Hayflick limit. And what's happening, your telomeres are the ends of your chromosomes. And they don't encode for genes, proteins. All that information is shown here in the light blue. And so this is like a cap. And over time, your telomeres get shorter and shorter and shorter until eventually, when they're too short, the cell undergoes apoptosis, which you should remember is programmed cell death. So this is a normal life cycle of a cell. It starts out as a stem cell, it differentiates into its specialized cell, it divides, and eventually it is signaled to undergo apoptosis. Okay. What's going on with some cancer cells is that they're not losing these ends, they're not losing their telomeres, and so they're never signaled to die, self-destruct. And so I put this figure up just to remind you what is the issue with telomeres. So DNA replication can only go 5' to 3'. And so if you remember your DNA replication, you use RNA primers, and when those RNA primers are removed and the gaps filled in, the very ends of the chromosomes of those strands of DNA can't be replaced because DNA polymerase cannot fill anything in here. It always needs a 3' hydroxyl. And so as you undergo rounds and rounds of DNA replication, you lose genetic material. Okay? And so that's what this is trying to illustrate. You lose these telomeres, which is fine. That's why they're there. They're there until your cell has divided enough times, and then it undergoes apoptosis. Well, there's an enzyme, telomerase, which makes the telomeres on the end of your chromosome. And when a cell has been specialized or is differentiating, telomerase is shut down. Okay, so we're no longer going to keep extending those. So this is active, the ends of your chromosomes. This is active in stem cells, not in differentiated cells. Okay. Telomerase can be reactivated in cancer, which makes the cancer cell what we call immortal, which means in theory it can live forever because it can continually build. This is telomerase building, extending that end of the chromosome, and so DNA replication may lose some, and then telomerase will put it back. So the cells can continually divide. They no longer have the Hayflick limit because they're fixing their ends. So it would be like you're stuck here. You're not losing your telomeres. So that's one characteristic of cancer cells, is they can have their telomerase activated. I apologize, I'm just getting over a cold. Another characteristic of normal cells is this thing called anchorage dependence and density dependent inhibition. So anchorage means cells don't like to divide unless they're sitting on some kind of solid thing, right? Like your extracellular matrix, okay, or other cell layers that they're supposed to be growing from. So anchorage dependent means cells require a surface for division, okay? Cancer cells can be anchorage independent, which means they can divide even when they are not sitting on a nice surface for cell division. Okay, so they could divide in your blood. Okay, that can be an issue. Density-dependent inhibition is cells normally form a single layer. And we see this in tissue culture, which is growing cells in the lab. And once they start touching each other, they give signals to each other to stop growing. Because if they start piling up on each other, they're not going to get the nutrients and the oxygen and get rid of waste like they need. So what they're trying to show you here is you could dig out a piece of this layer and the cells would then divide and fill in the gap. Okay? And once they filled in the gap, they would stop. Cancer cells don't have density-dependent inhibition, so they can continue to grow and they can pile on top of each other, and that's what's going to cause a tumor. Okay. All of this is controlled by cell signaling pathways, and I've just thrown up a cell signaling pathway. You don't need to know any of this. Okay, we've talked about EGFR. We'll talk about it later, but I want you to remember that the chapters we talked about, 22 and 23, about all the signal transduction, this is important because cells get information from their environment that says grow, divide, or it says stop. And what we're going to talk about in cancer is that a lot of times cancer is because cells are not responding to their signaling pathways. So they continue to divide. That's why this is a disease of cell cycle. Okay? Remember that normally there are these checkpoints that as your cell is going through the cell cycle, it stops, it pauses, it checks. Do I have all the growth factors, DNA damage cleaned up? Are we good to proceed for DNA synthesis? Okay, did DNA replication happen well? Has the cell got enough mitochondria and the cell size is okay? Okay, did the spindle fibers form? Are we ready to divide the chromosomes correctly? Well, what happens with cancer is that you're losing some of these checkpoints. And so instead of making sure the cell is good and healthy and ready to divide, it just keeps going through cell division, sometimes without even an outside signal telling it to divide.